my God. Do you see that?
In November 2021, a doctor managed to study Bigfoot's DNA up close, and he would be forced to cover up what he exposed for years. His discoveries would change what we know about the Sasquatch forever, with his results revealing a biological impossibility that shattered everything science claimed before. Dr. Alexander Burt and Jake Thornfield had been on their annual hunting trip in the remote Cascade Mountains, 40 miles from the nearest logging road. On their third day, they encountered a 9-foot-tall bipedal creature covered in dark hair that showed unmistakable intelligence in its movements and behavior. When the creature approached them, Jake's hunting instincts took over, and he fired a single-point 30-06 round, striking it in the upper chest. The creature roared and fled into the forest with impossible speed, but not before bleeding heavily at the scene. Jake's spent shell casing had landed in a pool of the creature's blood, creating a perfect sterile collection vessel. The blood was unlike anything Jake had seen in years of hunting, darker than normal mammalian blood, with an unusual viscous consistency that seemed to shimmer with an almost metallic quality under direct sunlight. Dr. Burt immediately recognized the scientific significance and carefully collected the sample, sealing it in a sterile container for analysis. Back at Pacific Northwest University, Dr. Burt faced the challenge of analyzing genetic material from a creature that officially didn't exist. He conducted the initial tests personally, working alone in his laboratory during off hours to avoid questions from colleagues and graduate students who might compromise the confidentiality of his research. The first microscopic examination of the blood sample revealed cellular structures that were simultaneously familiar and completely unprecedented in scientific literature. The red blood cells showed characteristics that were clearly mammalian, but their size and structure indicated adaptations that had never been documented in any known species. The cells were approximately 30% larger than normal human red blood cells, with a structure that suggested enhanced oxygen-carrying capacity that would allow sustained physical activity at levels impossible for normal mammals. The cellular morphology was unlike anything in the scientific literature. The red blood cells showed a biconcave disc shape similar to human cells, but with additional internal structures that appeared to be specialized organelles for enhanced oxygen storage and transport. The implications for the creature's cardiovascular and respiratory systems were staggering. These organelles were unlike anything documented in mammalian biology, suggesting adaptations for enhanced oxygen storage and transport that would allow the creature to function at high altitudes and during periods of extreme physical exertion without the fatigue limitations that affected normal mammals. But it was the blood typing analysis that delivered the first truly shocking discovery of Dr. Burt's investigation. When he attempted to determine the creature's blood type using standard ABO and RH factor testing protocols, the results defied everything known about mammalian blood classification systems. The blood sample showed characteristics that didn't match any of the known blood types in human or animal biology. The ABO testing revealed antigens that were completely unknown to medical science. Instead of the familiar A, B, AB, or O classifications, the creature's blood contained surface proteins that had never been documented in any terrestrial organism. The antigens were complex molecular structures that showed characteristics of both human and completely alien protein configurations, with binding sites that suggested compatibility with biological systems that didn't exist on Earth. Dr. Burt ran the blood typing tests multiple times using different equipment and reagents, each time confirming the same impossible results. The creature's blood contained antigens that were not present in any known blood classification system, representing a blood type that had never been discovered or documented in scientific literature. The antigenic properties were so unique that they would trigger massive immune reactions if introduced into any known mammalian circulatory system. The RH factor analysis was equally disturbing. Instead of the simple positive or negative classification found in normal mammalian blood, the creature's blood contained Rh proteins that were far more complex and sophisticated than anything found in known species. The Rh proteins showed characteristics that suggested enhanced immune function and resistance to blood-borne pathogens that would be lethal to normal mammals. The discovery of an entirely new blood type was unprecedented in modern medicine. Blood classification systems had been thoroughly mapped and documented for over a century, and the identification of a completely unknown blood type suggested the existence of a species that was outside the normal evolutionary tree of terrestrial life. The blood type characteristics indicated adaptations for biological environments that were fundamentally different from Earth's biosphere. Dr. Burt contacted colleagues at major medical centers and blood banks around the world, describing the unknown antigens without revealing their source. None of the leading hematologists had ever encountered anything similar, and the protein structures didn't match any known blood type variants or mutations documented in medical literature. The consensus among experts was that such a blood type was theoretically impossible within the framework of known mammalian biology. The hemoglobin analysis revealed protein structures that were fundamentally different from any known mammalian species. The oxygen carrying capacity was approximately 40% greater than normal human hemoglobin, with binding characteristics that would allow the creature to extract oxygen from air with efficiency that bordered on the supernatural. The hemoglobin variants showed adaptations that would function effectively at altitudes where normal humans would suffer from severe hypoxia. The unknown blood type included hemoglobin variants that were completely outside the normal classification system for oxygen-carrying proteins. The hemoglobin showed characteristics that would allow it to function in atmospheric conditions that would be toxic to normal mammals, suggesting adaptations for environments that didn't exist on Earth. The protein structure included binding sites for gases that were not present in Earth's atmosphere, indicating adaptations for completely alien environmental conditions. The white blood cell count was extraordinarily high, suggesting an immune system that was far more robust than anything found in humans or known primates. The cellular structure indicated resistance to diseases and environmental stresses that would be lethal to normal mammals, suggesting evolutionary adaptations to extreme conditions that went far beyond normal biological parameters. The white blood cells themselves showed characteristics that were completely unprecedented. They were larger and more complex than normal mammalian immune cells, with internal structures that suggested capabilities for fighting pathogens that didn't exist in known biological systems. The immune system represented by these cells would be capable of resisting diseases and toxins that would be fatal to normal mammals. The unknown blood type included immune system components that were completely unprecedented in terrestrial biology. The white blood cells contained proteins and cellular structures that would provide resistance to pathogens that didn't exist in Earth's biosphere, suggesting adaptations for environments with completely different disease vectors and biological threats. The immune cells showed capabilities for neutralizing chemical compounds that were not found in terrestrial biology. The lymphocyte analysis revealed cellular structures that indicated resistance to viral infections, bacterial pathogens, and environmental toxins that would be fatal to normal mammals. The immune system represented by these cells would be capable of resisting diseases that had decimated human populations throughout history, suggesting genetic adaptations that provided near immunity to most known pathogens. The platelet analysis showed clotting factors that were far more efficient than normal mammalian blood, with coagulation capabilities that would allow rapid healing from wounds that would be life-threatening to normal animals. The clotting mechanism included protein factors that were not found in any known species, suggesting adaptations that would allow the creature to survive injuries that would cause fatal blood loss in normal mammals. The coagulation cascade showed modifications that would allow blood clotting to occur at rates that were at least 5 times faster than normal mammalian blood. The enhanced clotting capabilities would provide protection against blood loss from injuries that would be fatal to normal animals while maintaining the flexibility needed to prevent dangerous blood clots during normal circulation. The unknown blood type posed questions about the creature's evolutionary origin that couldn't be answered with any known scientific framework. Blood types were among the most conservative genetic traits in mammalian evolution, changing very slowly over millions of years. The discovery of a completely unknown blood type suggested that the creature was either from a completely separate evolutionary lineage or had been artificially created using genetic engineering techniques that were far beyond current scientific capabilities. But it was the DNA analysis that revealed the true magnitude of what they had encountered in the forest. The initial genetic sequencing showed a genome that was approximately 96% identical to modern humans, closer than the relationship between humans and chimpanzees. But the remaining 4% contained genetic material that didn't match any known species in the global genetic databases. The foreign DNA sequences were complex and organized in ways that suggested artificial manipulation rather than natural evolutionary development. The unknown genetic sequences showed characteristics that suggested they were not the result of natural evolution. The complexity and organization of the foreign DNA indicated artificial manipulation or hybridization that was far beyond anything possible with current genetic engineering techniques. Dr. Burt ran the analysis multiple times using different equipment and techniques to verify his results. Each test confirmed the same impossible conclusion. The creature's DNA contained human genetic material that had been combined with something else, something that didn't exist in any known biological classification system. The human genetic component was not from any single population or ethnic group. Instead, it appeared to be a composite of genetic material from multiple human lineages, as if someone had deliberately selected the most advantageous traits from different populations and combined them into a single genome. The genetic diversity represented in the human component was far greater than what would be found in any natural population. The selection of human genetic traits showed obvious intelligence and purpose. The genetic material included sequences associated with enhanced physical strength, improved sensory capabilities, increased disease resistance, and superior cognitive function. Someone or something had deliberately chosen the best characteristics from multiple human populations and combined them into a single genetic blueprint. One of the most shocking discoveries came when Dr. Burt analyzed the creature's chromosome structure. Normal humans possess 46 chromosomes arranged in 23 pairs, but the creature's genetic material showed 48 chromosomes arranged in 24 pairs. The additional chromosome pair was unlike anything documented in mammalian biology. The extra chromosomes contained genetic sequences that were completely foreign to terrestrial life. These chromosomes appeared to be the source of the creature's enhanced capabilities, containing genetic instructions for biological functions that were theoretically impossible in normal mammals. The chromosome structure suggested that the creature was not simply a mutated human, but a hybrid organism that had been created through genetic manipulation that was far beyond current scientific understanding. The chromosome analysis revealed that the extra genetic material was not randomly inserted into the genome. Instead, it had been precisely integrated into the creature's genetic structure in a way that suggested sophisticated knowledge of genetic engineering. The integration was so seamless that it appeared to be the result of deliberate design rather than natural mutation or evolutionary development. The additional chromosomes contained genetic instructions for biological processes that were completely unknown to current science. The genetic sequences included instructions for cellular functions that would provide capabilities far beyond anything found in normal mammals, including enhanced physical performance, accelerated healing, and cognitive abilities that bordered on the supernatural. Dr. Burt's analysis of the creature's telomeres revealed another impossible characteristic. Telomeres are protective DNA sequences at the ends of chromosomes that shorten with age, serving as a biological clock that limits cellular lifespan. In normal humans, telomere length decreases over time, leading to cellular aging and eventual death. The creature's telomeres showed characteristics that defied everything known about cellular aging. Instead of the expected shortening that occurs with age, the creature's telomeres appeared to be self-renewing, maintaining their length through mechanisms that didn't exist in any known biological system. The telomere structure suggested that the creature possessed biological immortality with cellular aging processes that had been completely eliminated. The telomere analysis indicated that the creature's cells could divide indefinitely without the normal limitations imposed by cellular aging. This characteristic would allow the creature to maintain peak physical condition throughout its lifespan, with cellular repair and regeneration capabilities that were far beyond anything found in normal mammals. The genetic mechanisms responsible for telomere maintenance were completely unknown to current science. The creature's DNA contained genetic instructions for cellular processes that would prevent aging at the molecular level, suggesting knowledge of biology that was centuries ahead of current understanding. The telomere maintenance system included genetic instructions for enzyme production that would continuously repair and extend telomere length. The analysis of the creature's mitochondrial DNA revealed another shocking discovery. Mitochondria are cellular organelles responsible for energy production, and their DNA is typically inherited exclusively from the mother. The creature's mitochondrial DNA showed characteristics that were completely unprecedented in biological science. Instead of the normal circular DNA structure found in mammalian mitochondria, the creature's mitochondrial DNA was organized in complex three-dimensional structures that allowed for dramatically enhanced energy production. The mitochondrial modifications would provide the creature with energy generation capabilities that were at least 10 times greater than normal mammals. The enhanced mitochondrial function would explain the creature's extraordinary physical capabilities. The increased energy production would support the enhanced muscle performance, accelerated healing, and superior cognitive function that were encoded in the creature's nuclear DNA. The mitochondrial modifications represented a level of biological engineering that was far beyond current scientific understanding. The mitochondrial DNA also contained genetic sequences that didn't match any known terrestrial organism. The foreign genetic material appeared to be responsible for the enhanced energy production capabilities, suggesting that the creature's mitochondria had been artificially modified using genetic material from an unknown source. The mitochondrial modifications included genetic instructions for energy production pathways that were completely unknown to current biochemistry. The enhanced energy generation would allow the creature to maintain peak physical performance for extended periods without the fatigue limitations that affected normal mammals. The energy production efficiency was so advanced that it would revolutionize human understanding of cellular metabolism if it could be understood and replicated. Dr. Burt's analysis took an unexpected turn when he decided to run the human DNA component through genealogical databases to determine its geographic and ethnic origins. What he discovered made his hands tremble as he stared at the computer screen displaying results that should have been impossible. The genetic markers showed a direct familial relationship to Anna Penelope Doe, a woman who had lived in the Cascade Mountains region in the early 1900s. The mitochondrial DNA was a perfect match, indicating a direct maternal lineage that connected the creature to a woman who had been dead for nearly a century. Anna Penelope Doe had been a local legend in the small mountain communities that dotted the Cascade foothills. According to historical records that Dr. Burt painstakingly researched, she had been a reclusive woman who lived alone in a remote cabin deep in the same forest where Jake and Alexander had their encounter. Born in 1895, she had chosen a life of isolation that was unusual for women of her era, supporting herself through hunting, trapping, and occasional trading with the few settlers who ventured into the remote mountain valleys. The historical accounts described Anna as an unusually tall and strong woman, standing nearly 6 feet in height at a time when the average woman was considerably shorter. Local folklore suggested she possessed an almost supernatural ability to track animals and navigate the treacherous mountain terrain, even in conditions that would challenge experienced hunters and guides. Anna Penelope Doe had disappeared under mysterious circumstances, with her cabin found abandoned and signs of a violent struggle. Personal belongings were scattered throughout the structure, and the door had been torn from its hinges with a force that suggested something far stronger than a normal human had been responsible. Her body was never found, despite extensive searches of the surrounding forest. When Dr. Burt obtained Anna Penelope Doe's DNA profile from hair samples preserved by the local historical society, he made another shocking discovery. Anna Penelope Doe's blood type genetics showed she had possessed a rare blood type variant that was extremely uncommon in the general population, but showed partial compatibility with the unknown blood type found in the creature. Anna's blood type genetics included rare alleles that were found in less than 0.1% of the human population, suggesting that she had been specifically selected because of her unique blood chemistry. The rare blood type variants in Anna's genetics appeared to have been necessary for successful hybridization with the foreign genetic material. The blood type compatibility analysis revealed that Anna Penelope Doe's rare blood genetics had served as a bridge between normal human blood chemistry and the completely unknown blood type found in the creature. Her unique blood type had allowed for successful genetic integration that would have been impossible with normal human blood types. The discovery that Anna Penelope Doe had possessed rare blood type genetics that were essential for the hybridization process suggested that her selection had not been random. Someone or something had identified her unique blood chemistry and specifically targeted her because of her genetic compatibility with the foreign DNA. The genetic analysis revealed that Anna Penelope Doe's DNA had been combined with foreign genetic material through a process that demonstrated scientific knowledge and technological capabilities that were far beyond anything available to human science. The precision of the genetic integration was surgical in its accuracy, creating a stable hybrid genome that would be passed on to offspring without degradation or genetic instability. The creature's genetic structure showed evidence of multiple generations of controlled breeding and genetic refinement. Each generation had been systematically improved with enhanced integration of human and foreign genetic material. The breeding program had been conducted with scientific precision that demonstrated long-term planning and resources that were far beyond anything available to human science. Dr. Burt's analysis of the creature's protein synthesis mechanisms revealed another impossible characteristic. The creature's DNA contained genetic instructions for protein production that were completely outside the normal biological framework. The protein synthesis pathways were far more efficient than anything found in normal mammals, allowing for rapid tissue repair and enhanced physical performance. The creature's cells could produce proteins at rates that were at least 5 times faster than normal human cells, with accuracy levels that eliminated the normal errors that occur during protein synthesis. This enhanced protein production would allow the creature to maintain peak physical condition and recover from injuries at rates that would be impossible for normal mammals. The protein synthesis mechanisms included genetic instructions for producing proteins that didn't exist in any known biological system. These novel proteins were responsible for the creature's enhanced physical capabilities, providing structural support for the increased muscle mass and bone density that characterize the creature's physiology. The genetic pathways responsible for the enhanced protein synthesis were completely unknown to current science. The creature's DNA contained genetic instructions for cellular processes that would revolutionize medicine and biology if they could be understood and replicated. The analysis of the creature's enzyme production revealed another shocking discovery. The creature's DNA contained genetic instructions for producing enzymes that were completely unknown to biological science. These novel enzymes were responsible for metabolic processes that were far more efficient than anything found in normal mammals. The enhanced enzyme production would allow the creature to extract energy from food sources with efficiency that was at least 3 times greater than normal mammals. The creature could survive on minimal food intake while maintaining peak physical performance, suggesting metabolic capabilities that were adapted for extreme survival conditions. The novel enzymes also provided the creature with resistance to environmental toxins that would be lethal to normal mammals. The enzyme systems could break down and neutralize chemical compounds that didn't exist in normal biological detoxification pathways, suggesting adaptations for environments with completely different chemical compositions than Earth. The genetic mechanisms responsible for the enhanced enzyme production were completely outside the normal biological framework. The creature's DNA contained genetic instructions for biochemical processes that were decades ahead of current scientific understanding. The creature's immune system showed characteristics that were completely unprecedented in biological science. The immune system components were far more sophisticated than anything found in normal mammals, with capabilities for fighting pathogens and diseases that didn't exist in known biological systems. The creature's white blood cells contained genetic instructions for producing antibodies that could neutralize virtually any pathogen or toxin. The immune system showed adaptations that would provide resistance to biological weapons, chemical agents, and environmental hazards that would be fatal to normal mammals. The immune system also showed characteristics that suggested resistance to aging and cellular damage. The immune cells could identify and eliminate damaged or aging cells with efficiency that would maintain the creature's physical condition indefinitely. This capability would explain the creature's apparent biological immortality and resistance to normal aging processes. The genetic mechanisms responsible for the enhanced immune system were completely unknown to current medical science. The creature's DNA contained genetic instructions for immunological processes that would revolutionize medicine if they could be understood and applied to human health. The creature's sensory system genetics revealed capabilities that bordered on the supernatural. The genetic instructions for visual enhancement included adaptations that would allow the creature to see across electromagnetic spectrums that were completely invisible to normal mammals. The creature could detect infrared radiation, ultraviolet light, and other electromagnetic frequencies that would provide awareness of environmental conditions that were completely unavailable to normal animals. The visual enhancement genetics included genetic instructions for eye structures that could function effectively in near-total darkness, with retinal modifications that would allow the creature to see clearly in conditions where normal humans would be completely blind. The enhanced night vision capabilities would provide the creature with significant advantages during nocturnal activities. The auditory enhancement genetics were equally remarkable. The creature's DNA contained instructions for hearing capabilities that would allow detection of sounds across frequency ranges that extended far beyond normal mammalian hearing. The creature could detect infrasonic and ultrasonic frequencies that would provide awareness of seismic activity, weather patterns, and animal movements across vast distances. The olfactory enhancement genetics provided scent detection capabilities that were far beyond anything found in normal mammals. The creature could analyze chemical compositions with precision that rivaled sophisticated laboratory equipment, providing detailed information about environmental conditions, food sources, and potential threats through scent analysis alone. The enhanced olfactory capabilities included genetic instructions for nasal cavity modifications that would allow the creature to track individual humans across vast distances and identify specific individuals by scent alone, even days after they had passed through an area. The scent detection capabilities would provide information about the emotional state, health condition, and dietary habits of other organisms. The sensory enhancement genetics also included instructions for electromagnetic sensitivity that would allow the creature to detect electrical fields and magnetic variations. This capability would provide navigation abilities that were completely independent of visual or auditory cues, allowing the creature to navigate with perfect accuracy even in complete darkness or adverse weather conditions. The muscle fiber analysis revealed protein structures that could generate force levels far exceeding normal human capabilities. The creature's genetic code included instructions for muscle development that would allow it to lift weights and exert forces that would be impossible for normal mammals of comparable size. The muscle genetics suggested strength levels that were at least 10 times greater than those of normal humans. The muscle fiber composition was unlike anything in the scientific literature. The genetic instructions called for a combination of fast-twitch and slow-twitch muscle fibers that would provide both explosive power and sustained endurance. The creature would be capable of tremendous bursts of speed and strength while maintaining those capabilities over extended periods. The skeletal system genetics showed adaptations for supporting tremendous weight and absorbing impacts that would shatter normal bones. The creature's genetic blueprint included instructions for bone density and structure that would create a skeleton capable of supporting its massive frame while maintaining the flexibility needed for rapid movement through difficult terrain. The bone genetics included instructions for calcium and phosphorus deposition that would create bones with density levels far exceeding those of normal mammals. The skeletal structure would be capable of withstanding impacts and stresses that would be fatal to normal animals, while maintaining the flexibility needed for complex locomotion. The neurological genetics included instructions for brain development that would create cognitive capabilities, far exceeding those of normal humans. The genetic modifications suggested enhanced memory, improved pattern recognition, and problem-solving abilities that would make the creature one of the most intelligent beings on the planet. But the intelligence enhancements were combined with genetic instructions for predatory behaviors that were completely alien to human psychology. The creature's brain would be wired for hunting and killing with an efficiency that was purely instinctual, creating a being that could think like a human but hunt like the ultimate predator. The neurological modifications included enhancements to spatial reasoning that would allow the creature to navigate complex terrain with perfect accuracy, even in complete darkness. The genetic instructions for brain development would create cognitive mapping abilities that would make it nearly impossible for the creature to become lost or disoriented in any environment. The unknown blood type included specialized proteins that would support the enhanced brain functions, dramatically increased metabolic demands. The enhanced cognitive capabilities would require significantly more energy than normal brain function, and the blood chemistry modifications would provide the necessary glucose and oxygen delivery to support the creature's superior intelligence. Dr. Burt's comprehensive analysis revealed that the unknown blood type was not simply an incidental characteristic of the creature's biology. It was a sophisticated biological integration system that had been specifically designed to support all of the genetic enhancements encoded in the foreign DNA. The unknown blood type represented a level of biological engineering that was far beyond anything in current scientific understanding. The blood chemistry modifications had been designed to work in perfect harmony with all of the other genetic enhancements, creating a biological system that was far more sophisticated than anything found in terrestrial biology. Dr. Burt's comprehensive analysis led to conclusions that challenged everything he thought he knew about biology, genetics, and the limits of what was possible in the natural world. The creature represented a level of genetic engineering that was centuries ahead of current scientific capabilities, demonstrating knowledge and technology that didn't exist in any known scientific institution. The genetic evidence pointed to the involvement of beings whose identity and origin remained completely unknown, but whose capabilities far exceeded anything in human experience. The precision of the genetic modifications and the sophistication of the biological enhancements suggested intelligence that was far beyond current human understanding. After months of analysis, Dr. Burt made the difficult decision to keep his findings confidential. The genetic evidence was too extraordinary to be accepted by the scientific community, and publication would likely result in his research being dismissed as elaborate fraud rather than legitimate scientific discovery. He sealed the remaining blood samples in secure storage and encrypted his research data, knowing that the evidence documented discoveries that were decades ahead of their time. The Anna-Penelope Doe connection had transformed a cryptid encounter into evidence of genetic engineering capabilities that challenged everything humanity thought it knew about the limits of biological science. Some secrets, Dr. Burt realized, were too dangerous to share with a civilization that wasn't prepared for the truth about what level of biological sophistication had been achieved by beings whose identity and origin remained completely unknown.